Up next is Sarah Jobbins in the neuroscience program in Dr. Helen Lai's lab, and she'll be presenting her project, Pain Sensing Neurons, Assembly and Care Instructions Inside.
Although millions of people live with acute and chronic pain, there are very few effective and non-addictive treatment options available, partially because we don't fully understand how pain-sensing neurons are developed and maintained. This is the question my research tries to answer, with the aim of trying to help development of new pain therapies. To help you think about how we understand this, imagine that you're building some IKEA furniture, perhaps a desk and a couch. You'll probably want unique instruction manuals so you build them correctly, right? Now replace each piece of furniture with a unique class of sensory neuron and the manuals with unique gene combinations, their little hats here, and you'll have the basics of how we hypothesize sensory neurons are generated. In this example, the desk is a nociceptor, or pain-sensing neuron, and the couch is a proprioceptor, which provides our sense of limb and body position in space. We got a clue about how these sensory neurons obtain their identities from a disease called Puridium-12-associated congenital insensitivity to pain, in which patients have a mutation in a gene called Puridium-12 that results in their inability to feel pain and subsequently frequent injuries. When we recapitulate this in mice by removing Puridium-12 before birth, we see that pain neurons fail to develop and that the animals don't feel pain, indicating that Puridium-12 is critical for the development of pain-sensing neurons. However, proper maintenance of neuron identity is also important for proper neuron function. And so I wanted to know if Puridium-12 is also important for maintaining the identity of pain-sensing neurons into adulthood. To answer this, I removed Puridium-12 from adult neurons in mice after normal development has occurred, and then explored the expression of various genes that we use to identify specific types of neurons. In doing so, I found that we get expression of a particular gene that we use to identify proprioceptors appearing within pain-sensing neurons. So what does this mean? Essentially, under normal circumstances, Puridium-12 is actively preventing gene markers for other cell types from appearing within pain-sensing neurons, allowing them to maintain their unique identities. However, without Puridium-12, we get neurons that are a little confused because they have two different identity markers. It's as if someone were to hand you a manual that's half A and half B. You'll wind up building something that resembles half a desk and half a couch. This is especially interesting because it suggests that one gene can control both the development and the maintenance of neuron identity simply by determining whether it provides the assembly or the care instructions. Moving forward, we hope that understanding these developmental and maintenance processes will allow us to be able to grow and regenerate pain-sensing neurons in order to help provide new pain treatments. Thank you.